Voy a subir un poquito el computador porque lo veo como...
Bueno, ahora estamos en línea, así que estamos también en YouTube. Ok, empezá entonces.
Es que todos están escuchando esta conversa que tenemos.
Sí. Bueno, te estaba contando las cosas. Poné el to screen.
Leo, ¿lo pongo y arreglo ya?
No sé, espera, ¿tú dónde estás?
Pero es que Leo me arregla mis cosas.
O tú no tienes el permiso.
Si tengo permiso. ¿Cómo no va a tener permiso?
Aquí estás.
Hola, Nick.
Aplicación número 1, ahí estás. Oh,
Leo, ¿por qué se quedó callado?
¿Cómo?
Leo, ¿por qué está callado?
El tuyo.
Si quiero poner el puntito ese. Espera. Partimos, Leo no dice nada, está callado.
Sorry, Susana, I was just solving some issues. Okay, we are ready to go for the last part of the first day, and the next speaker is Susana Sanchez, Associate Professor in the Department of Polymers, the School of Science, Chemical Science, Universidad de Concepción, Chile. Susana is an expert in membrane biophysics and dynamics and fluorescent methods to address these biological questions. And in particular, she spent a lot of work addressing the protein interaction in regards of lipoproteins and the interaction of lipoproteins with membranes and the dynamics of cholesterol in membranes. In the past, she was LFD user coordinator for a long time, and she is master all the techniques that they developed there, FCS, FLIM, SGP, etc. She's a great collaborator. We did a lot of stuff during my time there at LFD, and I hope we can continue. He's a great friend, and I hope today you will enjoy. I really like this talk from Susana, which is related to the photon counting histogram and protein aggregation in regards to ribosomal proteins. So thanks, Susana, and I pass the word to you.
Okay, thank you, Leonel, and thank you for the invitation. Yeah. So this is a talk that we normally put in this course because it's a real nice example of a technique that is called circular scanning FCS. The person that is there is Alberto Garcia Marcos. He's from Spain, and he was a graduate student at that time who did all the work that I'm going to present. His boss was Juan Pedro Ballesta, and as a collaborator, we have here also Dave Jameson. So let's start for the biological question that these people have. They were working in Saccharomyces cerevisiae, and this structure that is here is the ribosome from this yeast, and it have two subunits, the 6DS and the 4DS. If we open this structure, and here, we will find this structure here that is called the stock or P0. And to P0, there are 4 proteins, acidic proteins that will bound when the ribosome is active. And they are called 1 alpha, 2 alpha, 1 beta, and 2 beta. So just to simplify this structure, we make it like this. So we will have a P0 and the 4 acidic protein bound. So the question this group has was, is the organization of these 4 proteins homogeneous in all the ribosomes, meaning all the ribosomes have the 4 proteins, or is it heterogeneous, meaning any 4 possibilities essentially with the different combination of proteins. Why that was important for them? Because you can imagine a population with ribosome with different acidic protein. And then if every of these ribosomes will translate different RNAs, they will create a pool according to the equilibrium between the different ribosomes. So that is a pretty nice way to regulate the pool of protein in the yeast. So the molecular biology approach they took was pretty insane. They did several combinations of mutant single and double labeled EGFPs, single and double labeled yeast. In some of them, they just label with EGFP the P0, that is one per ribosome. In other ones, they label the P0 and one of the acidic protein, they knock out the native one. So all the possible combination with the 4. And then some other ones, they didn't label the P0 and just the acidic protein was labeled, etc., etc. All the possible combination. So they arrived to the LFP with 25 yeast to work with. And the control, so 26. So the first question we want to ask, and this is the talk about, this is the application, I'm not going to talk about the other one, was to first resolve the main technical problem. Can we distinguish in vivo ribosome labels with one GFP and ribosome label with 2 GFP? So at that time we choose the experimental approach was to use circular scanning FCS, which is pretty nice technique for small things, also when they're mobile and when they're slow. So in the analysis will be molecular brightness and PCH analysis in the alive cells, in the alive yeast. If we could do that, we were looking for having a brightness of 1 in those ribosomes that have 1 GFP and a brightness of 2 in those ribosomes that have 2 GFPs. I will explain a little bit the circular scanning FCS because I think Enrico didn't explain this in the morning, but the scheme is pretty nice. Enrico explained that in point FCS, you have a point spread function and illumination volume where you have inside molecules, and molecules can go inside or outside, and then you will see the fluctuation of the intensity. Later you can analyze. Now, this is when you keep the laser fixed in a point, but you can also move the laser with the scanning mirror. And the simple shape to move the scanning mirror is in a circle. So let's suppose that we move in an orbit and it takes 1 millisecond to go from the origin, and come back to the point. And then let's also suppose that we take 6 points around this orbit. So we stop 6 times and we take FCS data. And then we go another, let's organize this point by location, right? So it will be separated by 0.17 milliseconds. And then we go another round. So after 1 millisecond, we have again a collection of points in the same location from 1 to 6. And we continue, blah, 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 blah. And then we have this organization that is called the carpet or hyperspace. And then the nice thing is that each of these columns will be the equivalent to an intensity FCS intensity trace. In this case, separate by 1 millisecond each. So we can analyze them by autocorrelation or by PCH and get all the parameters and all the parameters from the molecules that are creating the fluctuation in that volume in that particular position. So how many points do we sample in this circle? Well, we can do it two ways. We can do no overlapping PSF. And in that case, we will get a carpet where this one will be separate from this row, separate from this row, but we will get that. In our case, this will be okay. Or we can do overlapped PCF. PSF. And in this case was what Enrico was talking about before. You will have a spatial correlation information that you can analyze by comparing one point with the other. We just want to use one trace in each of the carpet box, 3 or 4 in each of the carpet. But we are not cross-correlating. We are not doing a spatial correlation. So the carpet is an ideal carpet with the data. We'll have in the x-coordinate the spatial information, and in the y-axis the temporal information. So we can get every column of this one, it will be an FCS trace, and we can analyze by autocorrelation or by PCH. We did both, but the analysis that I'm showing essentially at PCH, meaning brightness of molecule, brightness and number of molecules. This is the real cellular world. They don't look so nice. And look at these intensity, vertical intensity traces. They look all messy because the cells are moving. But we can always do the trending, which is a solution that we can talk later about how is the trending. That also is in the articles. I can reference to them. So essentially, you can have a nice autocorrelation function to fit and get your data. So let's go back to the yeast. So we need now to fix the yeast and to do the experiment. So what we did, first of all, we have to put them at room temperature. We didn't have at that time a stage with temperature, and also we didn't want to. We want to have them at 25, so they will not start reproducing. I mean, like dividing, right? And they will be more quiet. And also we put them in 1% agarose. These are low melting point agarose. That so we put it in a sandwich between a slide and a cover. Ready. And the cells, they have to, since this work takes for months, so you have to normalize your samples. So our normalization in this sense was the cells absorbance, mean scattering around 0.5 at 25. So we will take a scanning FCS, circular scanning FCS, and this is a picture of one of the yeast with the EGFP inside. Scanning FCS, here at all the parameters, you have to know every single parameter for later do the analysis. So the computer will give it to you, and you have to optimize that. So after that, we will construct the carpet, and then we will determine the brightness of the molecules and represent it in a data box chart like the one I'm presenting there, like population chart. Let's see what we have here. Here are the results. So this is the normalized brightness, and here are the two controls. I have to say that this work with scanning FCS and all the FCS in general, it requires probably more controls than experiments. But at the end, the data are pretty robust because of those controls. So here are the controls. We are going to work for months taking data for this type of experiment. So we need a way to normalize the data so we can compare them in time. So what we did every day, we will take one control of EGFP, so a yeast with EGFP alone expressed inside. That should always be the brightness of 1. So we will take it every day. And we will always take P0 label yeast that will also have a brightness of 1. So then all the samples taken that day will be comparable to these two controls, and then assign the brightness of 1 of that day, and then we can compare with the following day. I hope you understand that what I said. So here they are. Now we're comparing data from different days. So we have EGFP will have a brightness of 1. yeast containing just a P0 labeled with EGFP will have a brightness of 1. So then what happened with the other two? In this case, the 2 alpha was knocked out, and it was a GFP 2 alpha expressed. And as expected, and this one we were happy at that point too, they have a brightness of 1. When we remove the 1 beta and we add a label EGFP 1 beta, we have a brightness of 2. Has have to be precise, right? Plus minus. And then what happened when we add 2? The last one over here is P0 label. Sorry, we remove P0 and 1 beta, and we add P0 and 1 beta. And what we get, beautiful 2, around 2. And we remove P0 and 2 alpha, and we add P0 and 2 alpha, and we also get double. Now, there were some interesting cases too. For example, these two, that we remove 1 alpha and 1 beta, and we just add 1 beta labeled EGFP, and we got a brightness of 1, meaning that there is some specificity for 1 beta. 1 beta will probably just go in a specific site for 1 beta, but not in the one that it was occupied by 1 alpha. And in the case when we remove 2 alpha and 1 beta, and we add 2 alpha label, it occupied both. So it went up. Now, at that point, we were all happy with the result. We get the numbers we want. But when you work with these things that are so precise and so delicate, you have to go even further. So then the question is, what is the origin of these fluctuations that we are looking at? So the autocorrelation analysis gave us an average diffusion coefficient of 0.02 and 0.06 micrometers squared per second, which is super low. So what are the things that can be moving inside the yeast? And can we find out where we're looking? Can we refine this conclusion that what we are looking inside? So we can have free protein at high concentration, right? Because we also have the G0 pretty low. So then what happened with the free protein? The acidic protein GFP, I put some values here, have from 39 to 60 kilodalton. So the theoretical diffusion coefficient will be like 90 micrometers squared per second. Now the EGFP inside the cells, like Enrico said, is much lower, 20 micrometers squared per second. We never found any fast component. This is too fast for what we saw. So we don't think we have any high concentration protein inside. Now, they can also be polysomes that are really slow, and since there are so many, the G0 is pretty low. So, but what happened is that from our biologist collaborator, we have the cells at 25 degrees, and the oxygen inside that gel that we put it to be pretty low. So they're not supposed to be present there. We don't think we have polysome. So we do think we have free ribosomes, but the free ribosome, and we also give this part, this control, the theoretical diffusion coefficient for the free ribosome will be 12 micrometers squared per second. We purify the ribosome and we measure the diffusion coefficient, and it was 15 plus minus 2 micrometers squared per second. So predicted inside the cells, according to all the proteins that we have from before, it should be between 1 and 3 micrometers squared per second. But ours was much lower, 0.2 and 0.6. So what else can be there? So Enrico also talked about this part. These fluctuations that we are looking at, it can be two things. It can be a diffusion, but it means a structure, a slow structure that's going in and out of the illumination volume. but it can also be a binding process that it can go on and off. And we are just seeing the changes in the intensity. Can we differentiate these two processes? Yes, we can. The way of differentiating these two processes is to change the point spread function little by little and measure the apparent diffusion coefficient you get. If it's a diffusion process, the apparent diffusion coefficient will decrease with this precision. If this is a binding protein, the apparent diffusion coefficient will not change. So we did it. We took all the data and we reanalyzed. But it was easy. We didn't have to change the PSF because we do have the carpet. And in the carpet, we just take one line and then we add, we take one row and then we add one to each side. So we took 1 row, 3 row, 5 row, 7 row, 9 row. So we start increasing the size of the analysis. And then what we see with the P0, that nicely decrease. So it is a diffusion process for P0. And then we did it with all the other ones, but this is 2 of them. For the P0 label, we did 5 cells and 11 areas, and we do see a nice decrease. And this one is that is double label, 1 beta and 2 alpha. We also saw a nice decrease. So we are looking at the diffusion process. And clearly, well, it's not blinking, it's not free protein, it's not polysome, they're just the ribosomes flow inside the cells diffusing. So this is the conclusion. I think the time is fine. We are measuring the diffusion of the ribosome in vivo and the PCH analysis, and this is, let's say, the first one is the biological conclusion, but for us was also a nice conclusion from the technical point of view. The PCH analysis in vivo can distinguish between a species having 1 or 2 EGFP labeled protein. And I think that's the last one. Yep.
Thank you very much.
Lista, Leo.
Beautiful and perfect time. So, Susana will not be able to stay for the breakout room. So if you have questions, you should not miss the chance to ask questions now. So I think we are very good in time, so we can have probably 1 or 2 questions from the audience.
Can I have one question, Leonel? It's Enrico.
Yes, of
Que
course.
no lo veo.
Ah, bueno. Me tiene que ver para la pregunta.
Claro.
Ok, ahora no sé si me ves o no, pero
Sí
yo
lo
he
veo.
visto mi video. Escúchame.
Enrico, can you ask in English? Because there are some people.
Everything is turned on. I turned on my everything that I can. Okay, so
You have to turn off your Spanish. You have to speak in English.
Okay, I'm speaking English. Okay, so people have done, since the time we did the TIS analysis, people have done a lot of progress in expression of RNAs, and they believe that they understand how a cells or a tissue can work just by expression of RNA. Now, one question that we discussed when you were doing those experiments were, well, but what if the ribosomes are different? And then maybe they will synthesize protein in a different way. Yeah. Do you know if there is any? So I said, with the enormous data that today we have about transcript, essentially single cell transcript of everything, is there any evidence that a change in the ribosome will change the expression of proteins?
I have no clue, Enrico. I don't know. I didn't follow this. I was just remember that I was just involved in the experimental part. But I don't know if Ballesta continue working with this, and I think Alberto he just quit science. I think so. I don't know what they're doing in that, but I don't know. I don't have an answer for that. I wish I have it. I don't.
Mm-hmm. So the question is, there is no progress as far as you know of the relationship between expression of genes and then proteins synthesized.
It may be, but I don't know. I personally don't know.
Okay, that's enough.
Okay, any other question from the audience? Meanwhile, Enrico, I think you can turn on your presentation.
Oh, okay, so it's my turn now? Okay.
Because unfortunately, Laura Estrada will not be able to make it. She's is having some health issues. I hope she's okay and nothing severe happened to her. And Enrico will be presenting basically a couple of applications, one related to the diffusion to the nucleus, and you say also something related to the
IMSD. I will start with IMSD.
Okay, you will start with IMSD. Great. So, okay, so now you are on and you can go on, Enrico. Thank you.
Okay, I don't know. I'm on in my screen.
Yeah, absolutely. And you can share your screen.
Okay, I'm sharing now or not?
You are perfect. Yes, go ahead.
Okay, so today we talk a little bit about correlation and correlation along a line, and Susanna did a very nice presentation about that. So I will talk about something which is called image mean square displacement, and you will see that. So the mean square displacement generally is a method used in single particle tracking to figure it out the kind of motion of a particle. So let us see what is the basic idea in using the spatial-temporal correlation, so where space and times are together. So in the spatial-temporal correlation, we assume, is the basis of the equations, that given a probability to find the particle as a function of time, that probability will change as a function of time by two factors. One, which will increase the variance of the width of the Gaussian and will increase linearly with time. And the other is that the amplitude have to decrease too in order to maintain the integral to be a probability. And that will go with a more complicated rule, which is here. So the question is, if we can measure all that distribution and the distribution as a function of time, we will have much more information, and that is done by the IMSD technique. So let us summarize most of the technique I have discussed so far, and let us see if we can make a common point of all of it. So when we do single point correlation, we maintain the point, which is indicated by this blue circle, at a given time. And then if we consider we have a certain number of molecules, that time, as a function of time, the molecules will leave that volume, and then we will have, if we correlate that time with that time, or that time with that time, we will get here and here exactly the same, because the same molecules are in the same volume. But if we have from here to here, some molecules are left, so the correlation function will decrease. And then the farther we will go to the average value of molecules in a given volume, which is what we subtract, the famous one that we subtract in the equation. Okay, so this is what happens in the normal single point FCS, which is what I explained this morning. Now in scanning FCS or RIGS, what we do is that instead of maintaining the point in the same location, we move the point. You see, I am moving the point. And so by moving the point, two things can happen. Either we move the point very, very slowly, and then it's about the same point FCS, or we move the point fast, like Susanna was doing and so on. And then what happens is that we can have individual volumes, and then by moving the point, you can still study the distribution. So the distribution here, well, it starts from the same number of molecules, so it's supposed to be the same, but we are scanning the distribution. And in the spatial correlation, for example, at the single distance, so we have two volumes, so it's like the per correlation, we can see some of the molecules which are in one volume to go into the next. So those are the basic ideas shown in a graphical way of the methods we use. Okay, so I told you that if we measure EGFB in solution or EGFB in the cell, the correlation function stay the same, but it moves a lot, about a factor of 5. And people say that this is due to change in viscosity inside the cell. And I say this is not very convincing, because if you measure rotational rate of a molecule the size of GFP, you will find that actually they rotate exactly the same way. So let us try to join the different kind of thing. So we have essentially a Gaussian distribution, and this Gaussian distribution becomes flatter and flatter as we go with time. And if we have immobile part, so at some point it will change, but will not become 0, because we have some parts which are immobile. And that was discussed in the case of it. Okay, now let us see what is the difference between the IMSD, which stands for image mean square displacement, from FCS and RIGS. So let us consider in this column essentially the different process. So the process of a molecule which was originally in one side, it will move randomly and will explore a different side. So here is the molecule, but at this time, the molecule cannot go to the next side because it's confined, and we say that this molecule is confined. The other, which would be a transient confinement, which happens very frequently, is that the molecule is confined for some time and then jump over the barrier and can go farther. Okay, so let us see what the IMSD will measure, and I will show you the equation for that. So as a function of time, if we have pure diffusion, we will measure exactly what this expression was saying. So as a function of time, the Gaussian expression will increase linearly with time. Okay, so by changing linearly with time, we can see that the slope of the curve will depend on the diffusion coefficient. So the faster is the diffusion coefficient, per equal amount of time will go farther. And this is measured, so the IMSD is measured in micrometer square. And that is time, so micrometer square per second is a diffusion. Okay, and that works is, well, there are many experiments that show that works, and this is the simulations versus the theoretical values, and they are one-to-one. If you have confinement, what happens is the following, that the particle will start to move exactly in the same, but then cannot go as far as the diffusion will drive them, but they are confined. So, for example, if they are confined to stay in 400 nanometer, we will get that the maximum distance will be 400 nanometer. And then if it's 300, it will be 200. If it's 100, it will be 100. So we can measure the size of the confinement region just by measuring where the particle will go. And here is, again, a theoretical means what input to the simulation. This is what the simulation recover, and this absolutely real. Then we have a different kind of motion, which is a transient motion. So a transient means that for sometimes it will get confined, but then as the time goes on, it will go out of the confinement region, and then we will have like a free diffusion. And we can calculate that probability just by measuring the initial part and the final part. And so measuring all those quantities, which is available in a measurement of FCS, we can obtain all information about not only the diffusion, but also the confinement and transient confinement. So let us see how that appears in the, so the general equation for the IMSD function is an equation which depends on the coordinate x and x, well, this is in Greek, x and y, and the time. So this is the expression that we do. And then we have seen that this expression can be divided in different parts. So there is a G0 term, there is a excitation volume, so the volume of excitation, and then the probability to find the particle in the given position. According to Fick's law, this probability should be this expression, well, it depends on the dimension, but will be in this expression. So in the MSD, we divide the various parts. So we have that the correlation function will be a function of the temporal correlation. So if we stay at the same time, it will be just the diffusion. Then we'll broaden the probability by whatever is the fixed law, and then we'll have a constant offset. Actually, this is very interesting, and I will discuss what that means. Okay, so let us see the effect of the size of the particle. So there is an effect of the size of the particle, which means that when we measure the intensity of that particle, even if the particle can move at time t equal to 0, so the intercept of time t equal to 0, it will not be exactly 0 because it will be at least the size of the particle. So if we can measure that value, which is contained in this equation, so we can measure that value, then we should be able to measure the size of the particle. How precise is the measurement? Well, it's a lot precise. So if we do simulation again, if we have particles which are, this is 100 nanometers, this will be 10 nanometers, this is the recovered value. Well, it's more or less, follow in a line. So that means that between 1 micron and, 1,000 nanometer and 10 nanometer, more or less, we can recover the size of the particle. And this is very useful because that can give also information about size of clusters eventually. So I would say the particle size down to about 10 nanometer in this technique can be obtained with very high precision. Note that the sampling is low. A small particle will appear big. So if we expose a camera for some time and the particle is moving, the particle will appear big. So we need to sample as fast as we can in order to obtain this information. Okay, so let's go now to application of that. And the application of that is, there is a very interesting application, which is by the work of Kusumi, which is, he makes the hypothesis that in the membrane molecules, proteins will move, essentially just membrane protein will move depending on the cytoskeleton, and essentially there is a cytoskeleton, so the particle will move in a given part and will be transiently confined, and then eventually will jump over and then will go and more. So if you look at the data, so we have this data. So if we have essentially in the absence of lechunculin, which is a molecule that will destroy the cytoskeleton, we will have this behavior. So which means it's a transiently confined, as we have seen before. But if we have add lechunculin, then becomes almost free motion, and then we have free motion. So that was very nice, and you can obtain essentially the same result that we obtained years before by Kusumi. Okay, and then here we study as a function of temperature to show that this activation of a barrier, they didn't have this information, but we show that you can obtain an Arrhenius plot and calculate the probability to go over the barrier. Okay, now let's go to the important part I want to discuss here, which is this idea that you can do line scan faster and faster. And let us see what's up. Okay, so suppose you have a particle which is here, and then you scan over the particle. If the particle is not moving, you will see exactly a Gaussian, which will be the point spread function. So it's really how the point spread function is passing over the particle, which is immobile. But according to the rigs and everything that we see, if the particle is moving, you will have a narrow size of the correlation function. And then if the particle is moving, so the speed of scanning is much larger or smaller with respect to the movement of the particle, you will end up with something which is very, very narrow. So the line that we will measure will be will give us a direct information about the motion of the particle, about the diffusion of the particle. So we can do this experiment now because it's scanning. So we can sample at about a microsecond, and then we can go much lower And then we can measure as a function of the speed of scanning, for example, the diffusion of GFP in water, which is about 90 micrometers square per second. And you obtain this line. So don't forget this line, which will be in green in all the data that I will show. So the IMSD along a line shows that actually the GFP in water is a diffusion. In fact, is linear. And the slope is about what is supposed to be for IMSD. Okay, for the GFP. Okay, now let us see if we can do the same thing, but measuring in cells. So we can choose a part which is in the cytoplasm of the cell or a part which is in the nucleus of the cell. So that line green, I copy from the slope from the previous graph. So it's just what is supposed to be if we have a GFP in water. And this is in a cell now. And so notice that it deviates, but then it becomes like water. And becomes like water only if we can sample very fast. Otherwise, we will not see that value. And that is very rewarding because that shows that, yes, in a given a small size, and here is the size of the movement, so 10 to the minus 2 micrometers square or 10 to the minus 3, it moves like in water. Has exactly the same slope that the molecule is in water. So if you are a very small volume and which is the same thing, or very fast time, you will see that GFP moves like in water, which is what we always knew. Okay, now let us see what happens. Sorry, let us see what happens if we have. So remember, the green curve is just GFP in water, and now we are in the nucleus. Well, in the nucleus, we never get to be equal to water. Might be those points, but within there. But you see, it's always different, and so which means that it never reach in the nucleus. So this is the region we're exploring, the motion of like in water. Okay, so what is that, and where is that coming from? Well, we say, well, might be in the cytoplasm we have structures, so we have cavities, and so let us use a molecule like GFP. So remember, the green line is just GFP in water. So let us use GFP in this BSI solution, which is that point, and you can see that never changed the slope. So it's always the same. But, and then, so this is a solution, the fit, and this is in different amount of GFP. So the slope gives us the diffusion coefficient. So never change. But if we have a polymer, not BSA, but the polymer which has cavity which have water, and then polymer and water, then we can see this behavior in which we say that some region start to have the same slope than in water. And that size of the cavity corresponds to the physical size of the cavity we can measure with other means. So that means that the interior of the cell, at least the cytoplasm, is made of cavities where the GFP molecule can move very rapidly, like in water, which is the result we obtain from the rotational rate. And to give you an idea of how good is the system, so for example, this is Alexa 488, which has a diffusion coefficient of 428 in water. And yes, it moves exactly with the diffusion coefficient. You can measure very accurately those values. And this is for beads, which simply means that they are also diffusing. Hello? Yeah, which also means they are diffusing, and they are diffusing more or less with the same speed. So there was a question I had about Frap, what is the difference between Frap and the other techniques. So let us make this graph, and that's the last one of this presentation, in which I showed the region in which you can apply Frap. So at the best, you can go to 10 to the minus 10 to the minus 4 second, or I would say that it's more limited to that region. But anyway, but it's diffraction limited. So you can make a spot which is smaller than the diffraction limit. FCS is also diffraction limited, but the IMSD and RIGS is not. And you can go to much, much faster time, which also means much, much smaller distances. So look at movement in very small distance. So that shows another difference between the Frap technique and what you can measure with those spatiotemporal techniques, RIGS or IMSD or PCF and so on. So I will stop here for this presentation. I wanted to show this data because I think, what complement very well the discussion that we had when I give my lecture. It's like, where is that coming from? So there is nothing magic. Simply, it's the structure of the cytoplasm, which is different than the structure of the nucleoplasm, which is different, which is more similar to a polymer with cavities. Okay, so I will stop here and answer your question, and then go to the next application.
Thanks, Enrico. Very nice. I'm trying to see if there is any question in the chat, or if anybody want to.
Well, maybe I have other two things. I think this is a comparison with the technique that has state FCS, in which we can go slower, we can go smaller and smaller volume, and this is by IMSD. So more or less the points are very similar one to the other. So this is just. I have a question if the public doesn't have any question.
I have a question. If you think, you say, I mean, you need to go really, really fast in order to catch basically free molecule diffusing in
a small distance.
Well, this was my point. I mean, if you have a camera that you cannot run at that speed, how you can handle in order to get the very fast, like I don't know, fluorescing in solution?
Okay, so there is a different kind of technique which is limited, and this is the so-called case-space technique of Paul Weisman. Okay, so instead of measuring the correlation in the real space, like we are working, so our coordinate X and Y of the real space, you take the data, make a Fourier transform, and then you look at the various components of the Fourier transform of data. Okay, what happens is the following: that suppose you have a camera which is 512 pixels. So the minimum area that you can measure is 1 pixel.
Yep.
Okay. The maximum area is the size is 512 pixels.
It's the chip.
Well, it's the chip. It's the size of the chip. So you are very much limited because you can go a factor of 500 there.
Yeah.
And even if the camera is 1,000, generally you can go slower than the camera he was showing. But it's limited here. If you see the number of decays you have, so it's 1, 2, 3, 4. So essentially in the other region there is no limit. Yep. But you can never cross this line. So you stop at that point. And even if you use the space, you exchange the space for frequency using the spatial Fourier transform, still, you cannot explore that enormous change that you have here with the line.
Yeah. Yeah. What I was thinking was, I remember when we wrote the paper for the comparison between path correlation and IMSD, and you asked me to make a plot, which was, well, either you can, if you have something that is moving fast, either you have to scan fast or record as fast as you can, or you have to go as farther as you need in order to catch the diffusion. So the speed.
And this is what I said, with the camera which has 500 pixels, you cannot catch it. You never reach that. Yeah. It's the same question, just in a different way.
And the other thing is, yes, speaking about Paul Weisman approximation, maybe you can just stand a little bit different in between IMSD and the spatial correlation that he does, because basically instead of looking at the spreading of the point spread function, he looked at the shift of the point spread function in order to see the directionality of the flow, right?
Well, PCF of course gives you the directionality. Okay, but it's based on scanning, and the scanning is much faster. So the only technique which is similar is using the airy camera. And you say that this is like, I will call that a nano camera, which is okay. In fact, in that case, the frame rate is 1,000,000 frames per second.
Yeah.
So yes, at 1,000,000 frames per second, you are here. And you can measure that distance, and you can measure that very well. But you have to go to a different kind of camera and use the camera in a different way.
Yeah. Okay, great. I don't see any. Oh, Ernesto has a question.
So I have written 4 questions here. So you only discussed the first. Okay.
Right. Now, Ernesto.
Hey, Enrico, I just wanted to ask you whether you can more or less have an idea of these confinement spaces where you have in the cytosol for GFP, if you can have an idea of the size.
Yes.
Let's say, so you have to scan this length at this speed to get the diffusion of water.
I mentioned that. So when it becomes like water, this is your size of confinement. And your size is, well, this is square of the size, but will be 10 to the minus, well, it's about 50 nanometer, you see. So you have to take the square root of that number, okay?
Yeah, and well, having that, is it homogeneous all around the cell? All this?
No, not really. So
I mean, not saying within the nucleus, but within the cytosol.
No, it's not. It's not, because if you get the mitochondria, they are much. If you have to go far, if you get the ER or the other things, it's changing. But you know, you are measuring by the intersection of those 2 points. This is water, and this is interception. So that point of intercept is when the 2 things start to go together, and that happens in different positions. You are absolutely right.
But that's great, because you can say, okay, within these places you are more confined or less confinement. That's great, and with a determined pore size, let's say.
You get the average size, of course.
Exactly.
The size is the average.
Yeah, you also consider, Ernesto, that you are an averaging a region, because it has at least 32 or 64 pixel square.
Yeah, but the per correlation function, you don't have this limitation in some way, because you move the point.
Exactly.
So right. Here also, you have a line scan, but the line scan is extremely powerful, because you can go, count the order of magnitude, and in this side there is no limit. And I try to summarize in this graph, so that the normal technique and the FCS are here. But then when you use this line scan, you open up the space completely.
Totally.
Wednesday, Lorenzo will talk about this correlation within the nano camera.
With the nano camera, which is essentially the same idea, but instead of being a line, you have a surface.
Can I ask the last question? Did you try this line scan in the plasma membrane with dyes?
Yeah. What do you mean? I show, well, maybe. So this is in the plasma, cytoplasma.
At the plasma membrane.
In the nucleus, never goes.
No, at the plasma membrane. If you have
lipophilic
dyes to see, let's say, I don't know, probably you can label with gene 1 with fluorescent cholera toxin.
Yes, I know that. No, okay. We haven't finished the work. There's still a lot of things to be done. Yeah,
exactly. Well, I'm coming up with the experiments that I would love to do. Okay.
Yeah, no. So of course, the famous structure in the membrane are supposed to be very, very small and very, very fast. And we are, well, this is a limit in some way. We can go maybe another factor of 10.
Okay.
But we cannot go much lower simply because you don't get very many photons. But you can go, in terms of size, another factor of 10. And that's in speed, a factor of 10.
Okay, cool. Thanks a lot.
Okay. And we're doing that now. Lorenzo will give a talk in which we get not only, so this is just intensity, but we get lifetime and spectral information. So that will give much more information. But you're right, will be an ideal thing to study a membrane. Yeah, MSD is very easy to do because you can do in the TIRF instrument.
Okay, Enrico, let's move on to the last talk.
Okay, let me see if I can. So last talk was my presentation, application number 3. What is that? Okay, that was the one it was supposed to be.
You have to replace the screen that you are sharing.
Stop sharing and reshare.
Yep.
Okay. That one? Is that okay now?
Yes, perfect.
Well, I have to make it presenter. I have to go out of the presenter.
Yeah.
Okay, so now it's supposed to be better. Okay, so in this application, I want to talk about something which we published some years ago with Luca Lanzano and Francesco Gandarelli. And we published as a letter in Biophysical Journal because we were in a hurry. And I don't think has really a lot of public, but I want to mention something which is very important about correlation functions. Okay, so we already discussed all this stuff, and then I have the scanning FCS principle, so which was discussed by Susanna, and I will say a few more things about that. So Susanna mentioned that I will talk, but I didn't at that time, about scanning rapidly a line or a circle, which is faster, and then come back to the various points. And then she talked about dividing the data in the carpet and plotting the carpet and so on. And the other thing was to obtain, so how that is obtained, and then to obtain information about the diffusion of molecules. Okay, and so the question is now, okay, what kind of correlation function can we obtain from this kind of analysis? And so let's go to the point of the separation and the other point, and let's go to the problem we want to find. So she talked about being overlap or no overlap in the points, and both are important if you want to see very fast process. Okay, and this is, for example, when you have particles, and so that's part of our software, obtain all this stuff. But now let us discuss what happens for example, when we do the intensity at one particular column of this function. And on one function of this, we obtain what is just a normal correlation function that we will obtain if we have essentially a particle diffusing. And that is what she wanted to show. But actually, there is much more information here. And now let us open a different kind of application and see the information that we can obtain here. So somebody asked about if we are doing GUVs or other things. But GUVs move and, well, I don't want to discuss all the part. Okay, so let us start again with the nuclear pore complex. And now in the nuclear pore complex, we have some ternary complex made of importing alpha and importing beta that will transfer in the other side. And now the question is how the transfers occur. I show that NLS termination in the molecule makes a difference. But let us see what is the difference and how big is that difference. Okay, so first of all, how are made the nuclear pore complex? Well, the nuclear pore complex, we know most of the proteins. So there are 30 different polypeptides, so proteins, and each in multiple copies. And actually, it's an octameric structure. So there are 8 subunits of those many complexes which are acting all together. And they transport through a nuclear pore complex. You can transport ions, RNAs, of course, very important, proteins, entire ribosomes, etc. And to give you an idea, this is more or less in scale. So this part of the nuclear pore complex is about 50 nanometer, and this side is about the total size is about 150, 200 nanometer. All those are important things, and there are also important things to know that we know the proteins, we know the location, we know the octamer structure, and so on. So let us see what kind of things, well, we already discussed what kind of things can go through from the nucleus to the cytoplasm and so on. But let us see what we know about it. I jump to 10. So, jump to 10 again, involuntarily. Okay. Okay, so I want to understand how the transport through the nuclear pore complex occurs. So the passive transport, which is simply due to diffusion, is bidirectional, so occurs in both directions, independent, is regulated by molecular size. As I said this morning, well, was morning for me, about 60 kilodalton molecule can go through the nuclear pore complex, but it's energy independent, but it requires a lot of time, simply because the length of the channel is long. It's about 200 nanometers to 300 nanometers, and this is the time that is measured. So by diffusion, it takes about 150 milliseconds, and this is more or less the theoretical value. So active import is instead unidirectional, so can be from the nuclear, from the cytoplasm to the nucleus or reverse. Is not affected very much by molecular size, so we don't know how, but it's energy dependent, so it requires GFP. It requires ATP, sorry. Okay, so let us see how that occurs. So in the cytosol, there is a low concentration of NGDP. In the nucleus, there is a much larger concentration of NGDP, and this is the thermodynamic free energy that allows transport through the nuclear pore complex. In the cytoplasm, in order to get into this nucleus, you need to form at least a tertiary complex, actually much more complicated than that. And then there is some sort of interaction of that complex with proteins inside the nuclear pore complex, and then magically the system is, the molecule of the complex is found in the other side. I say magically because the mechanism that people have suggested is that there is known FG proteins, or rich in glycine and phenylalanine, that are in the interior part here of the nuclear pore complex. And the basic idea is that that net can be broken, and so the hole opens up, and then molecules can go through, but still by diffusion. Okay, so if we measure the time it will take to pass through, we can exclude model. So there are three kinds of models. So one is essentially the destruction of the hole. The second is the phase separation, and the third is the saturated phase model. And let us see how that will happen. So, so clearly we need to know each individual molecule, how it goes through the nuclear pore complex, and this is the most important thing. So I already showed that picture that shows that essentially you don't get any information about how you go from one point to another, other than the time it stays in the nuclear pore complex. Okay, so I discussed about the pair correlation function, so I will not repeat those things because I already discussed, and go directly to the problem. Okay, so, okay, is the question of how selective getting, which is one of the model for opening up the nuclear pore complex, occurs. And so we know that if the system here is highly disorganized, those proteins don't form any structure, and how it happens that it's open up. And it looks like, if you have to draw in 3D, it looks like really a spaghetti without sauce or something like that. Okay, so this is so what is the basis of this selectivity? How can distinguish that NLS is necessary to go on one side or not? And so the question is that there is an energy, a free energy, part is binding and part is entropic, and a molecule can reorganize the structure. But after reorganizing the structure, no matter what is done, it has to diffuse between one part and the other. And this is the point. So, so if you get the proteins which are inside, and then you can form a gel of those proteins, and then you can see that that gel at some given temperature, and depending on it can interact with some molecule and become like a fluid. And then they say, well, that has to be a mechanism, because that becomes a fluid, and so will allow molecules to pass through. And this is shown here. For example, you have a buffer side and the gel side, and then you add a protein which is marked with the DS red, and then you can see that after a very long time, everything you have in the buffer side will be found inside, and so on. So, and then based on that, they say that is the interaction with this protein that will break the bonds, and then eventually will form a liquid, and things will pass through. Another kind of model, which was proposed by Lim et al., says the following thing. So you have in the you have the GFP, the FG proteins, and then you approach with the AFM tip, which has this protein interact with the GFP, and you can see that as you get closer and closer, the extension of those proteins will change, and then essentially you form something which is from this side to the other side, and then you say, well, this is the process that occurs. But remember that the only thing you have done is simply decrease the size of those molecules. Still, in order to go from one part to the other, you need diffusion. And there are pictures that shows that, and so on. And so the question is diffusion versus direct motion proven or not? And the question is absolutely not, because all the diffusion, it depends on if you don't measure the time, you will never been able to figure it out if it's diffusion or it's active model. Okay, so let us see if we can start with a new technology. So here, so in green is importin beta. Sometimes it's called a caryophyrin because it's related to the motion. And this is the image that we obtained with my microscope that shows that actually some regions are enriched in importin beta. And then this is where a nuclear pore complex is, at least at this resolution. Okay, so the question is, can we focus on one of those nuclear pore complexes and see the molecules passing through? So, and how they pass through. So first of all, a nuclear pore complex in about 5 seconds moves very, very much. So you cannot do an image and figure it out which is which, because that shows that images at 0 seconds, at 5 seconds, and 9 seconds. So it's completely has moved completely out. So what we need to do is to track the nuclear pore complex. And so the fluctuation analysis will not give us very much because the objects move too much. Okay, so in order to do that, we will use the orbital tracking. I will not describe the orbital tracking, but the orbital tracking, you orbit a particle, in which case will be a nuclear pore, an entire nuclear pore complex. And as you orbit the particle, you will center the particle exactly at the center, in which case the intensity will be a constant. So the modulation, what we call this amplitude of the modulation, will go to 0. And this is the method we use. Okay, so let us see what happens. So if the particle is exactly at the center and we orbit around, the intensity will be constant. And so if it's not, the intensity will start to increase as the particle close to the orbit, and then we continue to increase up to about a certain factor. And then by measuring the modulation, we can feedback the system in order to bring the modulation equal to 0, which means we are centering exactly the particle. So this is in a few words how the method works. Okay, so now let us see. So we see the nuclear pore complex. So we make an orbit around the nuclear pore complex, and then we are look at this orbit. So in one side will be, for example, the cytoplasm, and the other part will be the nucleus. So any molecule that will pass through will appear in this part of the orbit first, and then will appear in the other part of the orbit first. So that was the basic idea. Okay, so let us see if we can go through. So now I draw essentially the same thing, but in a more realistic scale. So this is the orbit that we will do, and this is the nuclear pore complex in the way we see. So we are focusing on one, just one. This will more or less be the size of the point spread function. That will be the size of the nuclear pore complex. And we do the orbit with the time, which is 0.5 milliseconds per hour. So every 0.5 milliseconds, we do the complete orbit. So if the orbit has, we measure along the orbit, for example, 128 points, every point will be about a microsecond or something like that. So this is how the experiment will be done. Okay. So this indicates that we track, so the X and the Y move, but the modulation stay constant, and simply say that we can track it. Okay, now here is the point. So if we do a autocorrelation, just a normal correlation function, and we have the caryophyrin alpha and beta, which are marked, we never see a correlation function which is starting high and going low after a given amount of time. We see dots in the ACF carpet. And where are those dots? Actually, if we count, maybe we get 8, 1, 2, 3, 4, 5, I don't know, a certain number. And then if we analyze one of them, we see something very, very absolutely peculiar, that our autocorrelation function is made of a peak. We say a hump. And how an autocorrelation function can have a hump. And well, I said if it's diffusion, cannot have a hump, but if it's not diffusion, could have. And so let us see in the model what will be the idea. So the idea will be the following. So that sometimes, like this time and the blue, correlate always because always are at the same distance in time. But if other processes occur which are not at the same time, they will never correlate or will not give a hump. So, the idea is that a molecule is there, disappear and return there, and then something can happen in the middle, appears and disappear, appears and disappear, and this is the process that we're measuring. And since although it's random, it's not period, it's has a period, not in the total period, which does not exist, but has a period between the one position and the return at the same position. And this is very, very important. Now, that 2 points can be one side and the other side of the orbit. We have to figure it out now where they are. So we focus only on those processes which have an intensity and then an intensity at the given time. Remember that we are doing orbits. So how that can happen? Well, that can happen if a molecule, for example, while we are doing an orbit is here, and after some time is in the other side. And that time is fixed. Okay, and then even if the event occurs random, still will correlate and will give us a correlation which is, given by the time it takes a molecule to go from one side to the other. And if we continue, eventually things will come back, and then we can measure the time. So we can measure all those time independently, and we can make we can do the correlation in one direction, so point here and the point there, or a point there and the point here. So we can see the direction of the motion and the time it takes for that process to occur. Okay, so let us talk about the experiment, how we will do that. So we have a cargo that is supposed to pass through, has a GFB, which is green, and then the nuclear pore complex is in red or another color. So suppose we have the two molecules are independent. If they are independent, we will never see dots in the carpet because they are totally independent process, and then we will see only maybe some points with some correlation. So that means that there is more chances to see one passage to a given point of the nuclear pore complex and so on. But now, now suppose that we observe the passing of the cargo by making a cross correlation between the red part and the green part. If we make that, we will know if the cargo, which is in green, was here and then reappears in the other side, because it will change color, and then we can measure how long it takes to happen that. And so I say that is contrary to all the process. So we will have the whole process of diffusion, and then this PCF approach. So the correlation of the point along the orbit will give us the direction and will give us the time it takes for something which is in one side to appear in the other side. Okay. So, So now let us study this process. So we have the cargo, which is in green, and we have a protein which is just in red and is pure diffusion. So the green one will have a correlation, this hump in the curve, and that correlation time is 3 milliseconds. And the other one is much, much longer, which means, yes, it passed through, but at a total different time scale. Okay. So now let's go on with that part. And the question is, among the many proteins that we have, is there a protein in the nuclear pore complex which also move together with the cargo, which means some causality in the motion of the cargo? And so we choose a molecule which is NUP153, which is a very large protein, has 1,475 amino acids, is anchored near the N-terminal to the nuclear basket of the nuclear pore complex. So the basket is this part, so it's anchored here, and extend, contain a large number of repeat FG, and contain 4 RAN BP2 zinc fibers, and have both the N-terminal and C-terminal that we can mark with the EGFB. Okay, so we have, okay, so it goes back always. So we have a molecule that is anchored in the basket here, and eventually, if it's part of the transport, will go up to the other side and eventually contract again and bring the cargo in the other side. And that will be, we mark in green, and we can mark, say, the N and C terminal. And if we see the ACF, the correlation function of the NUP153 also has a bump. And it takes about 7.5 milliseconds to see so the characteristic bump is 7.5 milliseconds. So which means that every 7.5 milliseconds it goes up and then goes down. And so this is what is happening in this protein. So now we can do finally a cross correlation between the motion of the NUP153 GFB, so in green, and the cargo, which will be in any other color. And then we can see essentially when the protein is collapsed or when the protein is extended. Remember that the extension of the molecule is such the size that will go in the other side. So so the question is now, so we do the cross correlation, and then we observe what happens about the cargo, and what happens if the motion is correlated with the motion of the NUC 153. And essentially, we'll grab the cargo, eventually, some of the cargos, not all, of the cargo, and we'll bring back in the other side. And then we'll release, because here the concentration of the run GDP is very high. And then we'll eventually return, and so on. So now we can see in two colors what is happening in great detail about this stuff. And so we can measure the NUC 53 when it's collapsed, and so in the carpet. So every time we pass here, and once in a while it extends and then returns to the position. So this is the position in the orbit. We see that. So we see extending in 3.5 milliseconds. This is the altered region of active transform, and then eventually there is some inactive or passive transform, because the cargo can pass through the different region. So I think this is the first time that is shown that there is a correlation between the motion of one protein in the nuclear probe complex and the transmission of a cargo from one side to the other. And that motion occurs in the same time scale that we expect for active transport, and cross-correlate with the color, which is the cargo is colored down. So I think more or less this is the summary. So we present an approach based on the pair correlation function and the particle tracking, in which we can see the motion of a particle and cross-correlate the motion of the particle from one side to another of the nuclear probe complex. And this protein has this characteristic to be either compact or large or long. And that is not new. There is a lot of other examples in nature in which you have polymer can be extended or contracted, And okay, so that seems to be one of them. Why we choose nucleoporin 153? Because it's a long protein, because it's anchored in the basket, can extend up to the other side, and then come back. Of course, that does not explain that this is the motor that changes things, because there are many other things. So clearly, it cannot be a process based on phase separation or phase, because that will require too much time. So the cross correlation as a hint, like a spring-like mechanism, so extended or not extended or not extended. Okay. Now we did those experiments. He inhibited the NUP 153, and there is no more traffic, no more thing. But there is still a really large problem to solve, which is why the nuclear probe complex has 8 subunits. Are all 8 working independently, or are all 8 working all together to transfer things from one side to the other? And we don't know about that, because we mark only 1 out of the 8 nucleoporin, and might be, we should mark more of them and see cross correlation between the motion to answer that question. So clearly, we have made a step forward that we need this kind of pair correlation in order to see the motion of molecule. We can see the transfer of the cargo and many other things, but we don't know if that is really the mechanism or not. So I conclude right here, because I believe this is a really very interesting result about correlation function that can be have a peak instead of being just a relaxation like occurs in diffusion. So this is just to show that there are different kinds of correlation function, and then shows a possible mechanism for the transport to the nuclear probe complex. I finish here.
Thank you very much, Enrico.
Well, I want to thank Luca and Francesco.
They will be both on Wednesday.
Yeah, but they will talk about super resolution and about other kinds of things.
Yeah, I think this is one one of the most elegant work that has been done.
Oh, well, when
beautiful work.
yeah, I remember Francesco came to me and said, I see an autocorrelation function that has a hump. I say, so what? I say, impossible. I say, no, it is possible. So
yeah, it's really a beautiful work that
yeah.
they together, and I think it's very nice this combination of the particle tracking. Wednesday, Alex Valmishana will talk about that, and how the combination of particle tracking with the fluctuations and give chance to do all these things.
Yeah, and the fact that you can follow all the points of the orbit, and now today we can measure lifetime, we can measure Frap, so we can measure many other properties along the orbit.
Exactly. I do have a question. I don't see any hand rise or any question, but do you think, Enrico, if you, because you were talking and Nick was talking about the improvement that you can have if you have super resolution in the orbit. And I remember you have this idea of do orbital tracking with the STED. Do you think that change the picture?
Well, the center of mass, which is determined by the orbital tracking, does not need to have a super resolution because it's super resolved always.
Yes, exactly.
The center of mass is just, but if you want to see, for example, what other protons do around, then super resolution will help a lot. So, for example, how the proteins are organized or they move in those filaments that are intended to trap the molecule, and what happens in the reaction of release of the cargo, then you will need super resolution. But not for the orbital tracking per se is super resolved always.
Yeah, absolutely.
Because it's a center of mass.
Yeah. No, I was thinking about any kind of improvement in the correlation side.
It's in the imaging. It's done to have the imaging. But not the correlation, because the correlation, you have so many points at the center of those points. I have some images here about how those particles appear. They appear very, very sharp. I think there's some point here. Well, you see, those are the points, and this is one point in an orbit. So of course, those points are perfectly well defined. So you don't need much more than that. Because it's center of mass. So this is is particle tracking, which gives you the center of mass. And that was the discovery they did people 100 years ago, that if you can get the center of mass, you can get an enormous position in the position.
Yeah, exactly. Yeah, any other question from the audience? Let's keep some time. There is a question.
So we finish now today with the presentation, or there is more?
No, we finished today.
So we will have a breakout rooms, or?
No, because Susana is already left. So
okay.
The question that you wanna, well, the people can ask.
It's lunch time for me.
Okay, so there is a question here in the chat that says, studying the department diffusion in the nuclear envelope, can a hump appear if we have a line scan instead of a circular scan? So if you scan only with the line instead of...
Yeah, it could be done exactly the same thing, but I showed that the nuclear complex will move. So the tracking is done to track always one nuclear complex. If you do a line, you don't know which one you're tracking. So the reason why we do it with a circular thing is, first of all, you see both sides very clear, and then you know which side, but you can do that with the line. But if the nuclear complex move out of the line, well, what you do?
Yeah, this work, for example, with the model that Liz Hine was using to see the diffusion, just the diffusion of a molecule within the nucleus, like a green fluorescent with different oligomerization and this kind of thing. But it doesn't need to be fixed on the nuclear pore. So you
because she's looking everywhere.
Exactly.
But here, if you don't follow one, nuclear pop complex, you cannot really say what is happening.
Yep. Okay, well, if there is no more question before that we close the section and we leave, I will ask to all of you if you can turn on the camera. I would like to take a picture of the group. So, and then we can you stop sharing, Enrico?
I can stop sharing, so you don't want to have me in the picture.
No,
Oh, stop sharing.
I want to have you, not
No,
the
okay, I stop sharing.
Okay, so I am waiting just for all of you. It's funny because you can see different time in the time zone.
So I think we have 32 now participants.
Yeah, give me a second. I will try to take a print screen as soon all people are on. Okay, first page.
So you need 2 pages.
I need 2 pages, yes.
1, 2, 3, 4, 5, 5 times 5. So you have 25 now.
Okay.
So now you're going to go to the second page.
Perfect.
No,
Already,
you still have to do the second page.
I already did.
Oh, you did it. I didn't notice because I was in both.
You never notice.
I
Yeah,
didn't move out.
I already did it. Thank you very much to all of you for the first day. I hope you enjoy it. Tomorrow we will have another whole day focus on lifetime measurement, hyperspectral imaging, and phasors. I will open basically the Zoom probably 15 minutes ahead. So we will get in and be on time and try to be stick to the agenda. So thank you very much.
Okay, ciao.
All tomorrow. Bye-bye.
Okay.
Bye, everybody.